Hi everyone, welcome back to your channel Agriclarity and Clarity for sure. In today's video, we'll be discussing most important plant physiology questions which are very important for the upcoming IBPS AFO mains examination. Okay, so let's see why this session is important. So before moving on to the session, I would like to request every student watching this session to go to our channel Agriclarity and watch this PYQ analysis of plant physiology. Then only you'll be able to know which area is important to study in this examination. Okay. And these all analysis are done by the mentors who have already given this examination, who have cracked this examination. So you can trust on this analysis. And also we have made a separate PYQ analysis playlist on our channel where you can see that all the subjects which are very important for IBPS AFO examination are whose PYQ analysis we have conducted so far, like plant physiology, entomology, agricultural extension, horticulture, plant pathology, and so on and so forth. Okay, so I request everybody to watch this the PYQ analysis video first, then come on to this video so as to know the better the topics which are very important for this year's IBPS AFO examination. Okay. So let's move on to the first question of today's session. A farmer notices that in his maize crop, yellowing of older leaves first, while uh, younger leaves remain green. This is the most likely due to the deficiency of mobile nutrient. I have highlighted the key terms in this question so to be easy for you all to answer this question. Okay. Then which of the following elements could be responsible here? So the answer for this question would be answer option C, which is nitrogen, because what else are the mobile nutrients? Example, NPK, S, sulfur, and magnesium can move from older leaves to younger tissues. So deficiency symptoms appear in the older leaves first. Whereas if we go in detail about the nitrogen deficiency, it typically causes the chlorosis of older leaves. And in calcium and boron, which are immobile nutrients, their deficiency generally shows up in the younger leaves. Okay, and again iron and zinc, which are immobile nutrients, their symptoms show on the young tissues as well. Okay, so here we have discussed all about the mobile and immobile nutrients and their deficiency targeted parts of the plant. Okay. Next question is, which of the following statements are not correct regarding the essential mineral elements? Okay, so first statement says that it always be replaced by another chemically similar element. The requirement of an element must be direct or not indirect. Functional elements are always considered essential. And fourth is chlorine is considered functional as it can be substituted with bromine. So options are 1, 3, 2, 4, 1, 2, 1, 3, 4, and 2, 3, 4. So correct answer for this question would be A option, which signifies that first and third option are not correct regarding the essentiality or criteria of essentiality of the nutrients, which I feel that most of the students are familiar with the criteria of essentiality given by Arnon and Stout. Okay. So the first statement is wrong because the essential elements cannot be replaced. Second was correct because essentiality must be direct. Third was wrong because functional elements may or may not be essential. And fourth is again correct because chlorine is functional and can be substitutable by the bromine. Okay, that is why 1 and 3 option was not the correct regarding the essentiality of the minerals. Okay. Next is in a soil with excess phosphorus application, a farmer notices poor growth and leaf symptoms that resemble zinc deficiency. Okay, so which statement best explains this phenomenon? Here we are linking. We are linking the excess phosphorus application with the zinc deficiency. Okay, so the first option says that the P of phosphorus excess decreases the soil pH, making zinc unavailable. Then excess phosphorus excess increases the microbial activity, reducing the zinc absorption. Then excess reduces the root elongation, limiting the zinc uptake, and causes the chemical imbalances, inducing zinc deficiency. And last, causes boron toxicity, which marks the zinc absorption. From here, we have to pick out the most suitable answer. Okay, so the answer for this question would be option D, which is phosphorus excess causes chemical imbalances inducing zinc deficiency, right? So why? Because excess phosphorus causes a nutrients antagonism effect. Okay, that means a detrimental effect, or the opposite effect, okay, and leading to induced zinc deficiency, which is a classic case of hidden hunger, okay, where the zinc deficiency may appear, but despite adequate zinc levels. Hidden hunger means you can say that the means there is a sufficient application of zinc is given to that particular crop, but still it shows that it has the zinc deficiency problem going on, okay. So that for that case, it is known by the term hidden hunger. Okay. The fourth question of the today's session is which of the following statements about nitrogen and phosphorus deficiency are not correct? Here I am asking about the not correct statements. Okay. So the deficiency of nitrogen causes chlorosis of young leaves first, then while phosphorus deficiency causes chlorosis of older leaves. Nitrogen deficiency leads to purple coloration of stems, petioles, and veins due to anthocyanin accumulation. Okay. Then phosphorus deficiency may cause premature leaf fall and necrotic areas on the leaves or fruits. Then phosphorus deficiency can turn leaves dark green to bluish green or purplish. And lastly, nitrogen deficiency results in a stunted plant growth due to reduced protein content. So here we are discussing majorly about the nutrient deficiency and toxicity topic of the plant physiology. Okay. So the correct answer for this question is option A. Okay, where which is the not correct option regarding the nitrogen and phosphorus deficiency. Why? Because generally the nitrogen deficiency affects the older leaf first, because as we have discussed in the very first slide, that nitrogen is a mobile element. Okay, and the statement in option A wrongly reverses this. Okay, and all the other options are correct according to Fowler diagnosis, where nitrogen causes the anthocyanin accumulation, which was the correct diagnosis. Okay, and phosphorus causes the necrotic patches, premature leaf fall, and dark to blue, green, or purple leaves formation. Okay, so here we have discussed the foliar diagnosis as well. Now moving on to the question regarding the photoperiodism topic of the plant physiology. Okay, so a research student exposed that plants to alternating cycles of a short day followed by still shorter dark period. Okay, which of the following outcomes is the most expected? Okay, so here we are saying that when the plants are exposed to short day, then followed by still shorter dark day period. Okay, we are giving them dark day, but for a shorter period of time, then how will the plant react to this phenomenon? Okay, for example, in the case of rice, which is a short day plant, will flower normally. For the example of soybean, which is again a short day plant, which will initiate flowering faster. Wheat, which is a long day plant, will flower. Tomato, which is a day neutral plant, will not flower. And chrysanthemum, which is again short day flower, will flower early. So for the this question, the correct answer would be option C, which is uh, wheat, which is a long day flower, or which has a long day photoperiodism, will flower. Okay, so let's see. Short day day periodism require a continuous dark period. Okay, so uninterrupted. Hence, they fail to flower under alternating short day plus short dark day cycles. Okay, and if we talk about the long day periods photoperiodism phenomenon, where flower can under alternating cycles by followed by still shorter day dark period, as mentioned in the table, will still flower. Okay, so that is why wheat was the correct answer here. Okay, other options, as you can see, all the other options are generally short day. Then there is one option regarding day neutral, and again one short day. And the most appropriate example would be any example which is comes under the long day photoperiodism for this particular question. Okay. Let's move on to the next question. Which of the following pairs of element to function is correct? We have to choose the element and its function, which is incorrect in this question. Okay. So calcium, which stabilizes chromosome structure. Potassium, it has a function in regulating the water balance and stromatal movement. Sulfur, it is a component of coenzyme A and biotin. Iron, which is a direct constituent of chlorophyll molecule. And the magnesium, binding of ribosomal particles during the protein synthesis. Okay, so for this question, the correct answer would be option D. Okay, which is incorrect here. Rest options are correct. Okay, why? Let's see. So the first is the iron. if we say about the iron, it is essential for the chlorophyll synthesis, but not a direct constituent. Okay, so that is why we have mentioned that it is a direct constituent of a chlorophyll. So that is wrong. It is a essential for the chlorophyll synthesis because uh, there are other elements which are also involved in the chlorophyll molecule synthesis. Okay. Next question. We are then moving on to the topic growth analysis topic of the plant physiology. Okay, so we have to choose the correct pairs with their properties. Okay, like first is absolute growth rate. Its formula is given H2 minus H1 upon T2 minus T1. Then transcolational percentage indicates that estimate movement from grain to straw. Then harvest index, which is given by Nichipovich in the 1951, and the leaf area duration, sorry, which was given by Power and other scientists in the year 1967. Okay, so the correct pair for in this question would be option C, 1, 3, 4 only. Okay, second was wrong because the translocation percentage measures the source to sink, not straw to grain. Okay, that means straw to grain, not grain to straw. Okay, so it was reverse in this case. So that is why it was wrong. Rest options were correct. Okay. Let's move on to the next question. So eighth question of today's sessions is focused on the plant growth regulators. Okay. Here we are asking about the statement regarding the apical dominance, which is not correct. Okay. So the first statement is that it is controlled by cytokinins produced at the terminal buds. Okay. Auxin produced in the apical bud suppresses the lateral bud growth. Then third is removal of apical bud results in enhanced lateral bud growth. Then fourth is replacement of apical bud with agar block, which is a nutrient containing auxin, maintains suppression of lateral buds. Then last is Sook and Thiemann uh, demonstrated the role of cytokinins in the apical dominance. Okay, so the not correct options would be option B, consisting of first and fifth. Okay, so let's see why. First is wrong because apical dominance is not controlled by cytokinins; it is controlled by auxins. It is very clear from that first statement. So that is, it was very clear, and we can go for that. It is incorrect here. Okay. And the fifth statement, the role of auxins, not cytokinins, was shown by Sook and Thiemann. Okay, so here we are saying that the cytokinin role was demonstrated by Sook, but it was actually they were the ones who demonstrated the role of auxins. Okay, so that is why these two options were incorrect, and rest options are correct. Okay. Now moving on to the ninth question. Concerning the micronutrient deficiency, which of the following statements are not correct? Again, we are focusing on the not correct option here. So the zinc deficiency causes a little leaf disease where leaf size is reduced and stalks are short. Okay. Then the iron deficiency, intervariant chlorosis of older leaves because iron is mobile. Then manganese deficiency, molted chlorosis of young leaves with necrotic spots. Then copper deficiency, dieback of citrus and fruit trees, and boron deficiency, heart rot of beet, and brown heart of cabbage. Okay, so most of you must be familiar with the physiological disorders of the micronutrients. Okay, so the correct answer here would be option B, which is not correct regarding the iron deficiency, because iron is an immobile nutrient, and so the deficiency symptoms occur in the young leaves, which we have discussed earlier in the very first slide. Okay, and the statement B is incorrect because it describes that iron as a mobile, which is again it is an immobile nutrient. Okay, and other statements match correctly, as we have said that what all are the physiological deficiencies regarding these micronutrient deficiencies. Okay, like zinc little leaf problem, in the manganese molted chlorosis, then in copper dieback and white tip problem. Okay, and regarding the boron, heart rot and brown rot problem. Okay, moving on to the next question. Which of the following statements about the short day plants, okay, is not correct? Okay, so let's move on. Let me number them. Okay, 1, 2, 3, 4, and 5. Okay. So the first statement states that they require 8 to 10 hours of daylight and 14 to 16 hours of continuous dark period. Okay. Then we are considering the not correct options. Okay. So be careful here. Interruption of dark period with a red light prevents the flowering. Interruption of light period with red light prevents the flowering. Then propagation of, okay. Hmm. Sorry. Yes, propagation of dark period includes early flowering. Then maximum inhibition occurs if red light is given in the middle of the critical period. Okay, let me take one pen. Okay, given in the middle of the critical dark period. Okay, so the options which are incorrect in this question is option C, only three. Okay, why? Because other all first, second, fourth, and fifth are correct according to the given statement. And why the third was wrong? Because interruption of light period with red light has no inhibitory effect in short day plants. So only dark period interruption matters. Okay. So we in the third option we are saying that the light period is interrupted by the red light. Okay, which will prevent the fouling. And for that case, generally, it matters is that it is generally focused on the dark period. Okay, so they generally light period and red light has no inhibitory effects on each other. Okay, so that is why the third option was incorrect. Now moving on to the next 11th question, which are the following scenarios best demonstrates the commercial application of the cytokinins? So here we are talking about the plant growth hormones, commercial application. Okay. First is inducing seedlessness in the grapes in the vineyards, then breaking seed dormancy in lettuce seeds, then postponing the senescence and increasing the shelf life of fruits, then stimulating alpha amylase activity in the barley seeds, and lastly, promoting root initiation in stem cuttings. Okay, so the correct answer for this question would be option C. Okay, which is postponing the senescence and increasing the shelf life. Let's see why the other options are not correct. Where I mean, other options are also there, but most appropriate or most used application of the cytokinin is the postponing of senescence and increasing the shelf life of the fruits. Okay, so first, if we see go for the grapes, generally the grape seedlessness is due to gibberellins. Okay, then seed dormancy can be break through the usage of gibberellins and cytokinins, but mostly gibberellin is most prominent. Okay, then alpha amylase, again it is related to gibberellins, and rooting in cuttings is again related to auxin. Okay, so that is why other options were wrong, and the C option was the most correct fit here. Okay. Moving on to the statements, I have discussed what is I have given the general idea about the hidden hunger. So here we have to pick the statement which are not correct regarding the hidden hunger. Okay, so hidden hunger refers to nutrient insufficiency that shows that no visible symptoms but reduces the yield and quality of the particular commodity. Okay, then plant analysis and tissue test can detect hidden hunger. Hidden hunger has no impact on final yield or product quality. Potassium insufficiency in maize is a classic example of hidden hunger visible only at a maturity, and hidden hunger can result in abnormalities such as blossom and rot in the fruits. Okay, so here we have to pick the not correct answer. So the option C is the correct answer here. Okay, sorry, I have to erase it. Okay. Okay, so the option C is the not the correct fit here. Okay, why? Because hidden hunger does impact the yield and quality, and we are saying that it has no impact. Okay, so that is why it is incorrect. Even though visible symptoms may be absent, all other statements are correct. It is detected also. It can be also detected by tissue analysis, which we have mentioned in the second option. Okay, and potassium insufficiency maize is true that it is a classic example of hidden hunger, and it can cause yield and quality issues such as blossom and rot or poor fruit storage quality. Okay, so that is why the C option is the most appropriate option here. Next, moving on to the next question of the growth analysis part. Like net assimilation rate is different from crop growth rate in which of the following ways? So if you are clear with the definitions or concept related to net estimation rate and crop growth rate, this question would be very easy for you. Okay. So first question is that NAR measures per unit leaf area while CGR measures per unit ground area. Okay. NAR was proposed by Watson while CGR was proposed by Williams. CGR indicates that photosynthetic efficiency while NAR measures dry matter accumulation in whole field. NAR is expressed in gram per meter square per day, okay, and while CGR is expressed in GG per day per plant per day, okay. Then both NAR and CGR always show identical values for the given crop. So here we have to choose which statement correctly defines both NAR and CGR. Okay, if you have given any of you have given previous year IPS AFO examination, you have seen that many questions were there regarding the growth analysis part. Okay, from the plant physiology like leaf area index. Okay, net assimilation rate also. Okay. Okay, so that is why this topic is very important. So let's see what is the correct answer for this question. So option A is correct answer here. Okay. Why? Because NAR measures the dry matter increase per unit leaf area and it was given by Williams, not by Watson. And CGR, it also measures the dry matter per unit land area. Okay. And with per unit time. And it was given by Watson, not William, and thus the fundamental difference is that unit of measurement, leaf area and the land area here. Okay, so that is why in this way we differentiate both. Okay, net estimation rate is focused on the leaf area, while crop growth rate is focused on the land area. Okay, with the time taking into consideration. Next question. A farmer applied excess potassium fertilizer. Okay, so later his crop showed signs of leaf margin chlorosis and interveinal yellowing, similar to magnesium deficiency. So the what is the most logical explanation here? Okay, so potassium excess causes magnesium leaching from the soil. It directly damages the chlorophyll in the leaves. It induces the secondary magnesium and calcium deficiency. It reduces the nitrogen fixation in soil and it increases the aluminum toxicity. Okay. So the correct option for this question would be option C, which is potassium excess induces the secondary magnesium and calcium deficiency. Okay. Because again, they both have antagonism effect. Okay. Potassium in the soil has an antagonist which will antagonizes the magnesium and calcium uptake, leading to secondary nutrient deficiencies. This is why balanced nutrient fertilizer application is critical. Okay, so uh, this is it. Next question. A farmer notices his lettuce seeds fail to germinate in the darkness. Okay, we are again going with the plant growth hormone question. Okay, so you can see how diversified questions I have put. We are trying to give you targeting each and every topic of the plant physiology. Okay, so let's see. He treats them with a certain hormone and the seeds germinate vigorously even without the light. Which hormone mimics the effect of light in such light sensitive seeds? Okay, so options are IAA, indole 3 acetic acid, gibberellic acid, zeaxanthin, abscisic acid, NAA, which is naphthalene acetic acid. So the correct option here would be option B. Generally, gibberellic acid is used in lettuce and tobacco seeds which require light, particularly red light via phytochrome for germination. So this requirement is overcome by gibberellic acid, which substitutes for the light requirement here. Okay. So other options like auxin are mainly for the cell elongation and rooting, and zeaxanthin, which is a cytokinin, breaks seed dormancy but does not substitute light, and abscisic acid inhibits the seed germination. Okay, so if any of you watched our session on the plant hormones, you can see, go to our Agriclarity channel where I have made two separate videos regarding the plant growth hormones. Okay, in the first video, I have discussed the in detail with the biosynthesis analysis and all about the auxin and gibberellins. Okay, auxin and gibberellins, and in the second part of the video, I have discussed the rest left for plant growth hormones. Okay, so if you have studied that particular those two sessions and then I feel that this question would be very easy to answer. Okay, because I have discussed what are all the examples which were important regarding that particular plant growth hormone, how they are assessed, what is the significance of that particular hormone, how you can use them for the commercial application, which we have discussed in the earlier question in today's session. So you can see I have covered most of the topics here uh, on our YouTube channel. So please go and watch those channel sorry video, and I feel that these all questions I am giving you so far are of the at par level of the uh, actual IBPS AFO examination. If you feel so, please drop your suggestions or how you feel so far the questions are made for you, and is this questions are very similar to the pattern of actual examination. Okay, so I would like your suggestion, I would like to know your point of view for the session so far. Okay, so let's move on to the next question. So 16th question of today's session is that which of the following statements about the toxicity symptoms are not correct. So we are again moving on to the deficiency and toxic symptoms of the minerals. Okay, so excess potassium can result in magnesium and calcium deficiency. Excess phosphorus can cause a zinc deficiency. Excess zinc can lead to iron deficiency. Excess aluminum generally improves root growth and nutrient uptake. Okay, and toxicity of boron, chlorine, copper, manganese can directly cause visual symptoms in the crops. Okay, so the correct answer for this question would be we have to choose the not correct option. Sorry, not correct option. So the correct option would be D. Okay, which is the not correct. Why? Because excess aluminum does not improve the root growth. We are saying that it improves the root growth. It actually causes the root shunting, poor elongation of the root, and functional impairment. Okay, so the rest are correct, like calcium. Potassium can cause magnesium and calcium deficiency. Phosphorus can lead to zinc deficiency, and zinc can lead to iron deficiency. And again, boron, chlorine, copper, manganese can have a direct visual toxicity symptoms. Okay, so I request, apart from that, suggestions or your point of view regarding the session so far. If any of you feel that any topic is not clear to you or you want me to explain them in detail, you can also drop your suggestions in the comments below. I will answer your queries in the comments, and I will make sure that all the topics which you feel that you have not a proper grip on it, I will try to make a detailed and thorough video on it. Okay, so don't be shy away to drop your queries in the comments in the below in, in the comments below. Okay, so please, I would see all the comments and I will reply to all comments. Okay, next is now from here, so from the 17 to 20 question, we'll be discussing the assertion and reasoning based question, because we have seen that IPS AFO is also giving these kind of questions. Okay, so these questions will help you to assess your conceptual clarity. Okay. So first assertion is saying that cytokinins delay in leaf senescence by preventing chlorophyll degradation. The reason is that cytokinins enhances their RNA and protein synthesis in cells, thus prolonging the metabolic activity. The answer would be option A, because both are true and R is the correct, or we can say reason is the correct explanation of the A. Okay. So cytokinin does improve the RNA and protein synthesis, keeping chlorophyll intact, which is a Richmond-Lang effect. Okay. Next question of the assertion reasoning is that specific leaf area is a, again this question was a PYQ based question. Okay. So please carefully see the question. Specific leaf area is positively correlated with yield potential of a crop, and reason is that high SLA is always reflects efficient photosynthetic machinery in the plants. Okay, so the option for the correct option for this question would be option E. Both A and I mean both assertion and reasoning are false here. Why? Because the specific leaf area is a only ratio of leaf area to weight. Okay, and high SLA does not guarantee that a high yield would be there. The content specifically, or we can say the question specifically says that no relationship with the yield could be expected. Thus both the statements are false here. Okay, so I hope from this question the PYQ question based on the specific leaf area is clear for all of you. Okay, Next is 19. The assertion is saying that in short day plants, a brief exposure to red light during the critical dark period inhibits the flowering. Why the reason is that red light converts the PR, which is a uh, it is a phytochrome for the red light, and into PFR, which is phytochrome for the far red light absorption. Okay, so PR into PFR, and PFR form is inhibitory to flowering in short day plants. Okay, so the correct answer for this question would be option A, which is both are true, and reason is the correct reasoning, or reason for the assertion is correct. Okay, so red light does converts PR into PFR. Okay, and it accumulates PFR, which inhibits the flowering in short day plants. Okay, so from here we have discussed the what is necessary requirements for the short day plants, and what are the phytochrome involved in those short day plants. Okay, the PR and PFR are the phytochromes. Okay, for phytochrome red absorbing line and phytochrome far red absorbing line. Please, if you all know the wavelength of these two phytochromes, drop your answers in the comments below. Okay. Now, last question of the today's session. It is frost cracks in the tree, which is again a physiological disorder, occurs when the outer bark cools and contracts faster than the inner wood. So, reasoning for this assertion is that this differential contraction creates tension, causing longitudinal splitting of the trunks. Okay, so the correct answer would be option A, because both the options are true and reasoning is correct fit here. Okay, because frost cracks are a physiological disorder caused by sudden cold. Okay, there are, apart from the nutrient elements, there are also various physiological disorders like due to light stress, due to sun. Okay, due to cold temperature. So these are all important. We can say that biotic stresses. Okay, physiological disorder due to biotic stresses. So these are also important for the examination from the plant physiology point of view. Okay. So the mechanism exactly matches both assertion and reasoning, making the reasoning correct explanation here. Okay. So that was it for the today's session. You can check our AFO test series provided on our Clarity for Sure C4 Clarity for Sure courses. You can check the website link in the given in the description below. We have also given provided free all India mock test on the 7th September, and the reviews of the students and the reach of the students on that particular test was commendable. Everyone who have given that particular all India mock felt that it was an exact examination level. So please do check our that particular all India mock available on our website, and also you can go to our Telegram channel given in the that link is given in the comments in the description below, and you can check all the daily quizzes which we are posting daily on the 9 p.m., which are exact level of the IBPS AFO examination. Most of the new students who are giving test on that particular Telegram channel, they are feeling and they are already given good reviews regarding the questions. Okay, so please be sure that the questions given by our team are exact exam level of IBPS AFO mains examination, and all the mentors who have already prepared for this examination, have cracked this examination, are preparing wholeheartedly for you all. Okay, so I hope this session was very helpful for you all. Please drop your suggestions in the comments below how you felt this session was, any particular topic you want us to cover in detail regarding plant physiology or any part you feel, and we'll definitely reply to each comment which we'll receive. Okay, so that is it. Thank you, and stay safe and happy learning.